And thank you everyone for joining us today for the fourth and final webinar in our series on science and risk-based stability practices in pharmaceutical development. Today's topic will be regulatory landscape of science and risk-based stability. And as a reminder, today's webinar will be recorded and the slide deck will be made available to all registrants as well as the past webinars. All three of our speakers today work with the IQ Consortium Science and Risk-Based Stability Working Group and share an interest in using the science and risk and predictive techniques to stability to accelerate product development and focus stability knowledge and use throughout the product life cycles, as well as within their regulatory filing experience. Lisa Babayan is a principal research scientist in CMC Global Regulatory Affairs at Eli Lilly and Company. She provides strategic and tactical guidance for CMC development of small molecules by supporting global clinical trial and marketing registration submissions across diverse therapeutic areas. Prior to joining CMC-RA, Lisa spent 8 years as a research scientist in analytical sciences R&D, where she contributed to the development and support of solid oral and parenteral drugs. She received her PhD from Northwestern University and her BS from University of Southern California. Megan McMahon is a senior director in global CMC at Pfizer in Connecticut. Megan received her BS in chemistry from Purdue University and an MS in regulatory affairs quality assurance from Temple University. Megan started at Pfizer as an analytical chemist and has worked in regulatory CMC for the past 15 years. She co-leads Pfizer's Stability Council and is a member of Pfizer's Impurity Council. She also co-leads the IQ Science and Risk-Based Stability Working Group, which advocates for a holistic approach to stability development and retest period shelf life assignments, leveraging multiple stability tools and a stability risk assessment process to do so. Additionally, Megan co-leads the ISPE Patient-Centric Specification Working Group and was a founding member of the AAPS Chemical and Biological API Focus Group, where she served as chair for 2 years. And finally, Yan Wu is a director in analytical chemistry in development and supply at Merck with over 20 years of pharmaceutical industry experience. Throughout her career, she has served as an analytical lead on integrated development and supply teams for development of drug products in late clinical stages and support of new product filing and launch activities. She's led the stability project management, stability sample and data management groups that were responsible for stability study design and execution and filing for late stage development and commercialization of drug products, analytical development for drug substance throughout different clinical stages, and new product launch. She also served as a 2018 AAPS Stability Focus Group Stability Community Chair and is an active member of the IQ Risk-Based Predictive Stability Working Group. And with that, I turn it over to Lisa to kick off the webinar.
Good morning, everyone. Thank you, Brian, for the introduction. I will cover the introduction and hand it over to my colleagues, Megan and Jan, for the rest of the presentation. And we'd like to start the presentation with the mission and objective of our working group. If you had dialed into the previous webinars in this series, you're already familiar with these. But for those of you that are just joining in, the goal of our working group is to advance the application of science and risk-based stability approaches for pharmaceutical products through understanding the adoption status of science and risk-based approaches within the industry, understanding the existing roadblocks to successfully applying these SRB approaches, and we've provided tools and examples and also have advocated for regulatory change. As Brian mentioned, this is the final talk in the series, and hopefully you were able to attend all of the previous presentations that my colleagues have shared and have learned about the science and risk-based tools, their applications for both small molecules and biologics, and are ready to hear about the regulatory landscape, the success stories, and the ongoing advocacy efforts. In today's talk, I will cover the historical context of why this working group was formed and highlight the group's accomplishments. Megan and Jan will go over the current state of understanding of the regulatory acceptability of science and risk-based stability in the development phase, registration phase, and throughout the life cycle management. They will also highlight the current advocacy work and describe some case studies that will illustrate successful use of these science and risk-based stability strategies. Okay. So why advocate for science and risk-based stability strategies? And in order to answer that question, let's take a look at the background and the problem statement. So the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, which we commonly refer to as ICH, was formed in 1990. And the goal was to bring together regulatory authorities and pharmaceutical industry to discuss scientific and technical aspects of pharmaceuticals and develop relevant guidelines. Since the inception, ICH has evolved and now responds increasingly to global developments in the pharmaceutical sector. And these guidelines are currently being applied by a growing number of regulatory authorities. ICH's mission was to achieve greater harmonization worldwide to ensure that safe, effective, and high-quality medicines are developed, registered, and maintained in the most resource-efficient manner while maintaining high standards. The first ICH quality guideline to be published was ICH Q1A on stability testing of new drug substances and products. Q1A was followed by two annexes, Q1B on photostability testing of new drug substances and products, and Q1C on stability testing of new dosage forms. In 2003, there was a major revision to Q1A, which resulted in what is currently known as Q1A R2. And as part of that major revision to Q1A, additional guidelines like ICH Q1D, which talks about the bracketing and matrixing designs for stability testing of both drug substances and products, and Q1E, evaluation of stability data, were created. Q1F, that describes stability data package for registration application in climactic zones 3 and 4, was effective only for 3 years and was later withdrawn in collaboration with the World Health Organization. That definition for storage conditions for these climactic zones in respective regions was left to World Health Organization. One additional guideline was created in '95, and that was the stability testing of biological products. So over the past 20 years, Q1A R2 and its annexes have enabled harmonized approaches for conventional stability data packages to be used across global markets. However, the pharmaceutical industry has found that these conventional approaches can also present challenges in meeting the needs for fast access to life-saving medicines and for maintaining reliable supply chain. The science of stability has significantly evolved since the inception of these guidelines, and although Q1A R2 and its annexes clearly state that alternative approaches can be used with scientifically justifiable reasons, specific examples of acceptable approaches are not provided within these guidelines. They don't reflect the current tools and the strategies like stability risk assessment for risk-based predictive stability tools. And oftentimes this leads to unharmonized practices and mixed acceptance by regulators around the world. One good example is Q5C for biologics that does not explicitly include language for providing this flexibility to justify alternative approaches to doing stability. And there's a discrepancy between Q1A and Q5C that allows for this Q1A that allows for this justification, but Q5C does not spell it out, create confusion because biologics can be developed in line with the principles of both of these guidelines. As I mentioned on the previous slide, the current state of these guidelines does not reflect the harmonization that the ICH guidelines were intended to enable. And the challenges the industry has faced over the years can be attributed to different interpretations or understanding of the guidelines by regulators. Oftentimes we see with the regulatory feedback and the queries that the sponsors receive that there's the regulatory position that often is presented at conferences and the queries are not aligned are not aligned together. And also we have seen discrepancies within the same agency having different feedback when these approaches are used and justified. All of this discrepancy oftentimes leads to inefficient use of resources within the industry and is in contradiction with the mission to ICH, which I highlighted on an earlier slide, of developing and registering and maintaining medicines in most resource efficient manner while maintaining high standards. So one example of this misalignment of when regulators are applying guidances that are not aligned with the expectations outlined in the guidelines is the application of ICH stability guidelines for setting shelf life for clinical supplies, even though these are explicitly not within the scope of that guideline. So our vision is that an integrated approach should be applied to stability studies during molecules development and its life cycle management. As stability understanding needs changes throughout development, different strategies may be more suitable for different stability assessments. And by applying an integrated approach to using applied knowledge, risk-based predictive tools, modeling, we can appropriately address these different needs without relying on the traditional long-term stability, which currently remains as the regulatory expectations for all phases of development, from clinical all the way through life cycle management. Current regulatory environment indicates that there's significant global diversion regarding stability expectations throughout clinical development, registration cycle, and the post-approval life cycle management phases. And those discrepancies are with respect to the amount and the type of data that's needed. Oftentimes we see supplementary local requirements exist within certain countries that require additional information or specific requirements on where the data needs to be coming from, like site-specific stability. And with this being said, we've recently seen progress and movement towards adoption of alternative mechanism and incorporation of these flexible approaches. Specifically, we've seen this with respect to breakthrough medicine. And also recently we've seen endorsement for updating ICH Q1A and Q5C to align all of these expectations. So coming back to the mission of this working group, the working group was endorsed 6 years ago with the goal to achieve industry alignment on how to pursue these science and risk-based strategies to enable speed of delivering medicines to patients and helping drive global regulatory consensus on the acceptability of these SRB strategies. The group's accomplishments over the last few years are represented by the work that's resulted in numerous publications and presentations with the culmination of the latest publication shown here on the bottom of the screen for consideration of updating the ICH Q1A and Q5C guideline. Okay. So in the next few slides, we'll talk about regulatory expectations, advocacy, and experiences during the investigational phase with these science and risk-based approaches. There are changes and flexibilities that the industry would like to see in the clinical space of development, And those are the ability to implement changes based on accruing data that we're continuously collecting without the need of prior approval from regulators. The ability to set shelf life based on data that's not necessarily the long-term stability data that's currently expected by the regulators. And also we'd like to see some flexibility for extrapolating shelf life data based on previous knowledge and supporting information and where data supports that extrapolation. Some of the current roadblocks to all of this, achieving these flexibilities is specific recommendations and extrapolation limitations that are embedded within the guidelines. The divergence that we've seen on what data could be suitably be used to justify an alternative strategy. And the references to ICH guidelines and the data expectations imposed by these from regulators. We've already talked about the divergence, and I mentioned it on the previous slide. And just to highlight, on one hand, we have countries that do not have any requirements for commitments, for example, for shelf life. And on the other hand, we have countries that require an update to the shelf life to be filed prior to the implementation. And ideally, what we would like to see is a harmonized expectation and acceptance of science and risk-based approaches to setting and extrapolating clinical retest and shelf life. Oftentimes, we see that all of these divergencies result in increased time and cost that's required for developing a new product and can ultimately delay bringing this new medicine to patients. The IQ is working on advocacy, and the predictive tools coupled with the appropriate protocols can enable stability predictions within accelerated timeframes compared to more traditional ICH approaches. We want to advocate that we can use these tools to provide increased understanding of attributes that influence both drug substance and drug product stability instead of just following the traditional approaches that simply demonstrate stability in a very empirical manner. These modeling tools provide stability insights both within weeks, provide insights within weeks that could take months or even years to understand using long stability conditions only. And despite their usefulness, the knowledge that we've gained through these tools is not broadly used to support regulatory filing strategies. And what this working group is trying to do is to influence and to try to make the use of these tools and strategies to be more widely used and acceptable. Here are a few publications that demonstrate the prior work put out by the working group to support the use of these predictive tools for stability risk assessment and supportive data to drive the clinical stability strategies. I will pass the baton to Megan, and she will tell you about how these risk-based predictive stability tools have been used to support regulatory submissions, and we'll discuss the regulatory feedback that was received.
All right, thank you, Lisa. All right, here's a case study that I'd like to share with you. It's actually directly taken from one of our publications referenced below, and this publication is full of good case studies for your reference. In this case, it was a phase 1 clinical study where the company had some formulations already in the clinic with long-term stability on them, and a clinical shelf life had already been established at room temperature for 5 years for those strengths. They wanted to introduce a lower strength using a common granule, so really a low-risk situation from a stability perspective. But to support the introduction of that new strength without long-term stability study, they did kind of an ASAP study, predictive stability using a traditional kind of ASAP design using a crushed tablet to present the worst case from a degradation perspective. The results basically concluded that the stability profile for the new strength, as expected, was very comparable to the strengths they already had in the clinic, and they used that predictive stability data to support filing an amendment and introducing the new strength with a not a 5-year shelf life like they had for the already existing strengths, but a good 3-year shelf life to work with. You can see this was accepted in the US, France, Italy, Turkey, Egypt, Lebanon, and UK. For some reason I listed UK twice, but maybe that was because they can be difficult in this space. But it was a successful endeavor, and they started with a real good initial clinical use or shelf life for that strength. You can move on. I think we have a short poll for you guys now around whether, just very simple question, has your company used ASAP or other predictive tools to support an initial clinical shelf life without additional long-term stability data at the time of submission? So while you respond to that, I'll just note that the publication I referenced does discuss the use of predictive tools to set your initial clinical shelf life or clinical retest period without long-term data at the time of filing. And this is a really effective way of kind of speeding up the process of getting clinical studies moving. Often the longest data to come in is the long-term stability data, and if you're not waiting for that, then you can proceed faster. So it's good, and about a third of us have experience in the space. Okay, thank you. If we can move to the next slide. This is also directly taken from this publication, and the data is a little old. I think this is data through 2018. I know the IQ working group is working on kind of refreshing this data set, but this represents kind of the success of the authors of this paper. We collectively pooled our regulatory feedback, and the red bars here indicate the number the submissions that went to different countries that took this approach of kind of a predictive strategy to set initial shelf life without long-term data. So the red bars kind of represent the attempts at this strategy, and then the blue bars represent where this was successful without request for more data. So you can see there's a lot of success even in the early years. There are a few countries where this has never been successful, and these are areas where we try to influence those agencies moving forward. You can move on to the next slide. Here I'm going to pass it on to Yan to talk about our regulatory experiences in the registration phase.
Thank you, Megan. Can we move on to the next slide, please? So in terms of the current state of registration stability guidance, the ICH Q1, Q5C, that forms the basis for registration stability requirements in most countries. But as mentioned before, the industry has observed that country interpretation of these guidelines is variable. Individual countries may impose supplementary requirements to those recommended in ICH. So more recently, there's also this draft Pharm toolbox guidance that is available, that is on scientific elements and regulatory tools to support quality data package for Pharm marketing authorization applications. So this draft toolbox guidance embraces the science and the risk-based approach to stability, including using of supportive data, prior knowledge, modeling, and comparability studies. And also FDA has a guidance for industry that found expedited programs for serious conditions. So the guideline is broad, not specific to CMC, but does indicate that a flexibility regarding fulfillment to the ICH requirements may be considered. The next slide, please. So looking forward, we would like to advocate for tools recommended within the Pharm toolbox and to be universally accepted from acceptance through registration and life cycle. So for example, there's really no reason for clinical development strategies to be more conservative than the registration strategy. We also would like to encourage a holistic approach to stability and a stability understanding, including using the stability risk assessment, applying learnings from prior studies, and the use of predictive tools to assess the impact of changes. All of this really help us to remove the risk of surprises during the long-term studies. Next slide, please. So as mentioned, our IQ working group, we really would like to advocate for those changes. So most recent publication our group has is a white paper with the consideration for updates to ICH Q1 and the Q5C stability guidelines, embracing a current technology and the risk assessment strategy. So with that, we would really like to advocate for a holistic approach to stability understanding, utilizing historical data, prior knowledge, modeling, and the risk assessment process to expand the concept of what could be included or what would be acceptable in the core stability data package, including type and amount of stability evidence, an assignment of a retest period and the shelf life of a new product, and assessment of the impact of post-approval changes. Next slide, please. So here we just listed some of the recommendations we included into the white paper, and also a link to the publication itself if I have more time to go into more of the details. Once here highlighted here, for example, right, we would like to clarify that a representative batch for a drug substance could be made by different manufacturing process or even a synthetic routes that if you can appropriately justify through risk-based comparability studies. We also like to have an addition to a discussion or definition of the supportive stability and how supportive data can be used to inform a shelf life. Supportive data can include the long-term results from developmental studies and or through the statistical assessment, extrapolation, predictive studies, and or prior and platform knowledge. In terms of like the current pilot scale that was used in the current ICH, so that definition was more specific to the solid oral dosage form. So we'd like to propose to consider rebranding as a representative product, and with a definition that considers smaller volume products, diverse dosage forms, and continuous manufacturing. Next slide, please. So with our white paper, we also provided some recommendations that's beyond the core guideline for registration phase. So one of them is about risk assessment. So the recommendation for risk assessment process for it to apply to stability is consistent with ICH Q8 through 12. And it could provide the building blocks for companies to implement a science risk-based approach to stability. So this topic actually was covered more in details in the webinar 3. Another recommendation we have with the white paper is to have a stability modeling and or predictive stability and design and to be actually stated within the guideline. So the use of these tools provide increased understanding of stability performance and can reduce reliance on long-term stability. And these tools, they have the potential to actually reduce time to patient. They are also critical to enable the efficient decision point during development. Discussions on this topic were also covered in more details from webinars 1 and 2, and then there are slides and recording available to all the previous webinars. Next slide, please. So now we come to a second poll question. It's another short one. We would like to know if your company have actually applied predictive stability to support the registration commitments. So after this poll, we would like to share a few case studies to demonstrate how predictive stability can be leveraged to support the various sections of new product registration filings. So it looks like, about almost 30% have used, and that is encouraging. So I think there's also room for us to advocate more in this approach and apply this way of stability in the future. Thank you. Okay, so we would like to share a few case studies in terms of how predictive stability have been utilized successfully in the past to support various sections of the new product application. So the first case study showing that DOE-based predictive stability study design can be used to establish the regime of assured product stability and to support formulation development. And you can see from the diagram here that we were able to design the stability study with the different temperature and the moisture to really provide the control space within which that you can justify the product development for the section of P2.2. The next case study we have here, which is to the upper right, is how we can use the study to support the manufacturing process development, which is the P3.5, PP 3.2.3 in this case. So you can see here is the profile of the dissolution profiles of the API at various relative humidity. So the study was used to explore the impact of temperature and the moisture of the product to the dissolution and the chemical stability. And the results showed that an increase in moisture correlated to the faster rate of dissolution, and that this data could be leveraged for support of the manufacturing process development. The third case study we have included in here is actually by leveraging the predictive stability for a container closure system, which is a part of the P2.4. So this example actually was given in webinar 1, so I wasn't going into too much detail, but the output of the study can help really to provide a stability profile and a water activity threshold that can be used to recommend the primary packages for your commercial products, and this is a great way to justify it. The last case study listed here is the use of the study to justify specifications and the stability summary and the conclusions. That's part of the P5.6 and the P8.1. So in this case, we have a study that is performed to assess the impact of moisture and temperature on the stability of the API in an extended release tablet formulation over the anticipated 2 years of shelf life. So the study actually revealed that the API was sensitive to the moisture and the hydrolysis degradation was the primary degradation pathway. Through the analysis, the data revealed that the acceptance of product stability was observed in samples stored under the 35% RH. And it provided justification for proposed shelf specification limit of the hydrolysis product to be the 0.3%, which in the case of the formal stability study batches, we actually had a non-reportable hydrolysis product. So the product was filed worldwide, and there was no pushback to this approach. So you can show that this predictive stability results can really help to enable us to set more meaningful specifications and to support the shelf life, in addition to the regular formal stability studies. So with that, I will turn over back to Megan to speak to the regulatory expectations, advocacy, and experience for the lifecycle management.
Excellent, thank you, Yan. You can move to the next slide. All right, so in the post-approval space, one of the biggest issues, I think, is just regulatory divergence. We see requirements to implement changes vary extremely, and though that it has a downstream effect on impacting timelines to implement changes. If you're waiting for regulatory boards of health that require more stability data than others. An example we talk about sometimes is, the stability requirements to support a pretty simple API change can vary from no stability data needed to support that change to months of drug product data with the updated API in the new drug product to support that change. So you can see it's just filing those changes with that supportive data can be over a year difference, and then waiting for the approvals even longer. ICH Q12 does take a step towards trying to address harmonization in the post-approval world. They recognize that Q12 recognizes that Q1 and Q5C were not intended to be reused to assess every change. The intent of the recommendations in Q1 and Q5C are to set shelf life or to set retest period, and not necessarily, those don't necessarily need to be reestablished after a change. Really, the study to evaluate the impact of a change on product stability can be fit for purpose and can be done in many different ways. We as a working group are advocating, again, that white paper we'll reference speaks specifically to what we'd like to see addressed for post-approval changes. You can go to the next slide, please. So we did take a poll as a working group a few years ago, and this was the overwhelming response was yes, the industry would love to have some harmonization in the post-approval space. We basically asked, you know, would industry like some ICH or harmonized guidelines around supporting stability data to support post-approval changes, and the blue bars represent those who say yes, we definitely want that, and there was the green represents those who abstained. Nobody disagreed with this proposal. Can move to the next slide. Thanks. So Jan spoke to some of the recommendations that we put forth in this paper around our recommendations for Q1 and Q5C. The third annex that we recommended was one specifically that addresses post-approval changes, and it's really meant to kind of extrapolate or expand upon the ideas presented in Section 9 of Q12. That speaks very clearly to the appropriateness of using science and risk-based strategies to assess stability, the impact on stability that post-approval change might have. Next slide, please. This is an excerpt from the paper. So we have the paper is loaded with annexes and specific ideas, and this is just one of them. But here you can see there's kind of a two bucket approach that we're advocating for. On the right is kind of the situation where the company would take an approach of doing a thorough risk assessment around the impact of the change. And in the case of the right bucket, the company would have determined that either through the risk assessment or maybe they did a very short-term accelerated comparative study to demonstrate that there's no risk to stability, and therefore they could file their change with this justification and move forward without a stability commitment. On the left side, you have a couple situations falling into this bucket. So perhaps that company who did that risk assessment found there is something that needs to be further investigated from a stability perspective, in which case they might design the appropriate stability study to do that and include that in their submission or a commitment to complete that as part of their submission. It also in this portion or bucket, you might have the companies that don't have those SRA processes in place and want to follow a more traditional path and just generate the supportive stability that is kind of more standard at this time. Next slide, please. All right, here's one case study. We have a couple case studies in the life cycle space, and this is the first. So this is a situation where a company filed an NDA or a marketing application. In this case, it was an NDA for a complex modified release solid oral dosage form. There are multiple strengths, and they're all made from a common pellet. The primary stability data at the time supported a 36-month shelf life. The proposal that this company made was to kind of propose a reduced annual batch commitment for this product. The commitment was reduced based on a couple things. I think reduction in quality attributes that would be tested, but also the primary thing was they proposed to only test one batch of any strength per year. There were multiple strengths filed. They were all made from a common granule. The justification for this was based on their historical data around the data they had that supported that the formulation was pretty much equivalent across the strengths and performed equivalently, like the dissolution didn't change based on the strength of the dosage form, and there were no strength-related trends observed for any quality attribute. So based on that, the strategy was accepted and was successful. At this point, I just want to do one more poll, and this is the last poll, and this is around whether your company has used development knowledge or historical data to support a reduction of future stability commitments. Ready? Oh, here we go. Oh, yes. So that's actually higher than I expected. 50% have done this. That's really interesting. Okay, thank you. Our last case study is around a bottle change, and as we all know, packaging is often very important for stability of a product. And this bottle change was proposed by a company, and it wasn't just for a single product. It was a comprehensive bottle change for the whole commercial portfolio. So over 50 products were impacted, and within those 50 products, there were multiple presentations of each. So different strengths for each product, or maybe different bottle configurations, bottle sizes, bottle counts. So this was a bottle change that was going to affect a large number of products and presentations. The bottle resin that was proposed had not currently been used for any approved cedar product as well, which was another factor. The stability risk assessment indicated that this was a low risk change. The bottle was assessed thoroughly and for MVTR testing and other standard tests to ensure it would be suitable for protection. Next slide, please. So the approach that the company took was a risk-based approach with like kind of a bracketing approach, where there was a lead product selected, and that lead product the change was supported for that lead product with 3 months of stability data, which would be submitted via a CBE 30 in the US. This enabled the establishment of the bottle with cedar, and then and demonstrated that it was as protective as the other bottle. And then the remaining 50 plus products and all their orientations would be implemented via annual report with only one batch for each product. So even if the batch had multiple presentations, just the commitment was just to do one batch only. In addition, with the lead product, the supportive data for the bottle was submitted within the P24 section of the dossier that included the MVTR information and all the compendial compliance data that was generated. You can move to the next slide. So for those of us regulatory colleagues out there, how did we do this? Like from a regulatory perspective, this was done, I think, via a comparability protocol. This where the strategy was proposed to the agency via a comparability protocol that was submitted as a prior approval supplement. And once that was approved, the lead product was activated, and that was filed via a CBE 30, as I said before. And after that, then the remainder of the portfolio rolled out the change, and it was implemented via, and then notifications were provided via annual reports. Okay, and then moving into the conclusions, you can move to the next slide and discussion. So yeah, so in kind of to wrap up everything we've discussed here today, I think Lisa touched on this at the beginning, but when in 2005, when the publication of ICH Q8 came out, ICH began to address this important concept of science and risk-based approaches. And now there's a whole slew of guidances that kind of embrace this, and Q8 through Q9 provide guidance to industry on development, manufacturing, and life cycle management. But just to touch again on Q1 and Q5C, the original intent of these guidances was to kind of drive harmonization, and they did, and they really did a fantastic job of that. In addition, they actually did introduce science and risk-based approaches without really labeling them as such. And you can see this within the guidance around how to select batches and storage conditions, matrixing and bracketing strategies, and extrapolation of data. Those are all science and risk-based approaches. But as Lisa mentioned at the beginning, there's also reference to alternative approaches, which opens the door to other strategies, but without kind of examples of those, it's hard to have harmonized acceptance of such alternative approaches. So we are, of course, advocating for more guidance on those non-conventional strategies or science and risk-based approaches to further drive harmonization. Next slide, please. And the end goal, yeah. So I think if you've been to the other webinars, you might have seen this nice figure on the right before. But this kind of it represents what we're going for. Ultimately, we want to use science and risk-based approaches to further enable flexibility during development and over the life cycle of a product. The figure on the right, represents all the sources of stability knowledge that a company has. The gray thing at the bottom, hexagon, sorry, I had to count the sides, represents, what's currently really the standard approach to setting a shelf life for a product or a retest period, and that is long-term data and your traditional accelerated data. But in reality, the knowledge base for us is way broader than that, and we would like to, bring all that into the tool set that we use to set our shelf life or retest period, or to implement to support a change after approval. All of that is relevant and should be part of the assessment. All right, next slide. All right, finally, so I have the pleasure not only of wrapping up this specific webinar, but I think the whole series. The takeaways, I think, for this whole series is that industry's already moved towards a more science and risk-based approach to stability. We use it all the time during development to make very important decisions. The next step is using that knowledge base to not just drive development decisions and scientific development, but also regulatory strategy. And some of the companies, a lot of the companies that are members of our IQ group already do this. And so the key is really working with regulatory agencies to help them understand the value of these strategies that we're advocating for. They might fall outside the conventional stability that's more that they're used to, but building an understanding of these tools is the way to drive regulatory acceptance. And the members of this working group, we do this every day. I think, especially, us in the regulatory space, we do it through our companies and our regulatory submissions, where we propose different strategies, and we also do it obviously through the work we've shared with you today, all the presentations and publications, and this webinar series. And I think that is the last slide. But we have some time, about 5 minutes, for a couple questions. Thank you.
Excellent. Thank you, Megan, and thank you as also Lisa and Jan. So we do have some questions that came in. One of the questions is regarding case study number 1, which I think you presented, Megan. Was a commitment to station new strengths on stability submitted as part of the amendment?
Sorry, I muted myself. Yes, there was a commitment to do long-term stability, but there was no long-term stability included in the submission.
Okay. And then another question that's come in around some of the case or any case studies for applying RBPS approaches to biologics in a regulatory filing to leverage longer shelf life. So do we have any experience or have we seen that in our travels?
Yeah, we're at a disadvantage here. I think it just so happens the folks who put this final webinar together, we are all small molecule folks. And so I don't, we can put that on FAQ question because we can connect with some of our other colleagues and maybe address that in FAQ.
Yes, I think that's a great approach. And just to remind everyone, we will be putting a FAQ sheet together with all the questions that we don't have a chance to answer throughout the entire webinar series. 2 more questions just came in, or 3. Have you had any recent success submitting predictive stability approaches within the EU?
Sure, I think so. I think we, especially in the clinical space, that slide that I presented with the bar graph, there are a lot of EU countries on there that have accepted that approach.
Around the future efforts, how are we going to align with some of the markets which are not major markets and currently are not harmonized with the ICH? Do we have a plan for going in and trying to influence there?
Yeah, that's a good question. I think I don't have a solid answer for there. I think we start we start with ICH because that's the most influential space to start, and if we can make progress there, you know, I think the next step are countries that are non-ICH. But I think even countries that are not formally ICH members, like our strategies currently don't change drastically from country to country.
Okay. Another question around a market-specific question. Do we have any successful case studies using lean stability in China?
Oh, probably. I mean, I don't know. I can't. The question's a little vague. I guess when we say lean stability, that could mean a lot of different things. I know. I'm sure we've had experiences where if our stability commitments might for like post-approval stability commitments, where there's been a reduced protocol. I'm pretty sure we've had experience with that through the folks at IQ. I can't think of a specific example that I could speak to.
Okay, and that's something else that we could probably add to the FAQ and seek out from the larger team. So one last question with the last minute that we do have. Do we have any case studies that involve a drug substance source change while the drug product submission was at a pre-approval review stage?
Sorry, could we repeat that again? The drug substance source change.
Yes, any case studies that involve a drug substance source change while the drug product submission was at a pre-approval review stage?
No, nothing that I can think of.
Okay. Excellent. Well, thank you very much for attending, everyone, and thank you guys very much for a great webinar today. And hopefully you enjoyed today's webinar in the series, and Mahalea will make sure that we have the recordings and slides sent out to the to the registrants. Thank you very much and have a good rest of your day.
Thank you.
Thank you.